Thank you. Well, yes, Sydney said I don't do public speaking that often. I think this is the second time in about 2 years or 2 or 3 years. Anyway, what am I going to talk about? Well, I'm not going to talk about just my lab's work. I'm going to try to give you some, actually talk about what interests me in cancer, where I think the frontiers of cancer and genomics are, and maybe some insight into the level of thinking and expertise that's required to carry out sort of the next generation of molecular medical developments. And my lab, as Sydney says, works on autism. We do work in genomics, and there's always been an interest in cancer. Not so much at the therapeutic end, well, I'll talk a little bit about that, but more in terms of how can we improve prognosis and staging of the cancer and detection of the cancer, and I'll try to explain why those are extremely valuable tools to be able to develop. I'm going to try to give a broad talk with not very much in the way of technical details, but a high level of conceptual stuff. Groan if it gets to be too much. Although my emphasis is going to be on detecting cancer, it's not going to be the only thing that I emphasize. So Genomics of Cancer begins with the promise that it can be helpful in treatment and risk assessment and in detection. But the genomics of cancer depends almost entirely on the fact that cancers are mutagenic engines. They mutate at a high rate, and those mutations, to a large extent, drive the cancer. And much of cancer research, including our own early work, was dedicated to what are the critical mutations that make cancer malignant. The rationale for that is that if you can identify the genes that make the cancer malignant and inhibit those genes, or if they're genes that are lost that would otherwise serve as the brakes for cancer, somehow one can replace those, that one could treat cancer. And this is the thesis that's been going on since I started work in the field. That was mid-1970s. And the yield from that approach has been definitely in the positive column. There are a number of drugs. Herceptin is the most well-known. There are a number of others that are useful in leukemia, to some extent in melanomas and a variety of cancers, where drugs that target specific genes that mutate are useful in treating cancers. But it's still a vast minority that are treatable in this manner. And nevertheless, with the evolution of high-speed DNA sequencing, a lot of publicity has been given and a lot of attention and has been given to taking the cancer genome, sequencing it, and trying to find those mutations that are critical. Now, there's a problem with doing this. First of all, cancers have so many mutations that being able to pick out the ones that are critical is intrinsically very difficult. Second, cancers can have multiple critical mutations. In fact, in the early days, we were astounded to find what we called oncogenes, that many of the established cancers had multiple of these oncogenes activated. So it wasn't just turning on a pathway once, but turning on what looked like the same pathway multiple times, which is always, remains somewhat of a puzzle. So there are possibly multiple functionally significant ones. They're still in a minority. And to further complicate it, the cancer, as growing in a patient, isn't just one monolithic entity. The cancers are themselves heterogeneous. So you have multiple subclones, each of which may have different sensitivities growing in the patient. So the problem of treating cancers by treating the mutations, while it has had positive results, is still a hard road to go down, and it's one which, going back about 10 years, we decided to abandon and focus instead on all of the mutations that just are there, leveraging those mutations in ways that can improve cancer treatment. So the first slide, do I control that? Yeah. It's just, it's probably not necessary to this audience, but I'll do it anyway. I'm going to be talking about mutations. Let's specify what kind of things these are. So most common type that you find in cancer are point mutations, where you have some sequence of genome, and one of the nucleotides changes. So let's say the C gets converted to a G, and the complementary strand follows, and now you have a new sequence. That's the most common, point mutations. But there are many other things, other mutations that we call structural changes in the genome, among them things like inversions, where a portion of the genome just flips. So that's designated here. The arrows indicate the direction. This indicates a flip. And whenever these occur, they create a bridging fragment. That is a fragment that is not seen in the normal genome. Just like this point mutation is not seen in the normal genome, this bridging fragment is not seen in the normal genome. Or structural changes that we call copy number, and I've illustrated a copy number loss or deletion here, where this sequence just has a whole chunk that gets deleted out of the cell, and it too creates a bridging fragment. It has now a DNA sequence that stretches across this breakage point, and this bridging fragment is unique. So the mutations create things that in principle are uniquely detectable in the cancer and not present in the normal. And these are the guys that I'm going to be talking about, and discuss how they are useful. Okay. So, I'm not following my own script. I'd like to illustrate a couple of the things that I said, and this is work from our lab. So I spoke before about cancers being heterogeneous. This is work that was started by a graduate student, Nick Navin. I hope he's not calling. It works like this. We take the tissue. We prepare nuclei from the tissue. We sort them. We make what are called libraries so we can sequence them. And they're sorted into as single cells per well. Each library comes from a single cell, and we can sequence that genome from that cell and look at what I called, I've already introduced to you the concept of copy number. So this is a genome of one cell that has deletions here, 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 as you go from chromosome 1 to the end. So this cell has a series of marker events that distinguish it. And then we aggregate over the cells of the cancer, and when we do that, and we line them up by their similarity, you can see that they fall into what we call a phylogenetic tree. There's a branch of the cancer here, 2 branches here, and a 4th branch here. And their relationship to each other can be distinguished. We view these cells in something we call a single cell viewer. Each column is a single cell. So when you see a stripe like that, this is a common event in all of the cells. This is an event in a different set of cells. That's not common with these. So this is a heterogeneous cancer. And to bring that into a crisper focus, what we have here is what we call a heat diagram. And what is designated on the x-axis are each of the individual breaks that we can monitor in cells. And these are the individual cells. These are the cancer cells. These are the normal cells. You see that the normal cells do not contain these events. These are the events that are found largely in the cancer only. And the different clones have different patterns of events. So these events are present in all of the cancer, These are present only in these cells. These are present only in those cells. These are present in these cells. This only has the original mutations, and this one has the original mutations plus this. Now, roughly speaking, you could argue that the number of mutations of this type falling into a given cluster represents a period of sort of quiescence for the cancer, when it is plateaued in its growth, and then it breaks out by accumulating either enough mutations or just the right mutation to form another growing arm of the cancer. And if time is dependent, if time is proportional to the number of events, this clone, for example, has spent maybe 2/3 of its life in this stage and 1/3 of its life in that stage, and similarly across the way. So these could represent clones with very different properties because they've had very different experiences along the way. So to expect them all to respond to drugs in the same fashion is probably naive. We don't know yet how important this is in the resistance of cancer to therapy, but most people are strongly suspicious that it does account for a lot of therapeutic resistance. That you can be lucky with a combination of drugs to hit one clone, but you're not going to be necessarily hitting them all. So I'm going to stay on that theme. So we're still on the theme of genomics and therapy. And this little cartoon describes a cancer growing, let's say, in a breast. And I've designated the cancer as this stellate-looking body with arms, because cancers don't grow in a ball, they actually spread out. And I've designated sort of what the surgeon does when they remove the cancer. I'll be using this slide quite a bit. I'll come back to it. One of the very recent advances in cancer biology, and this isn't work at all related to my own work or those in my groups, but Dave Tuvison here at the lab is one of the practitioners of this, is that you can take the cells out of this cancer and grow them outside the body, and the efficiencies, and they grow in little things called organoids. And the efficiency of this process is quite astounding. So from one cancer, you can get many of these organoids. And so each organoid is derived from a single cell. So they will have the population structure. For example, these would all be represented in organoids. And unlike clinical trials on a patient, you don't get an opportunity to test 12 different drugs on a patient. And then you would need hundreds of patients to be sure of anything. You can do your clinical trials on these guys. And this is in the class of, I think, technologies that are going to revolutionize and accelerate the developments of therapies. So all my caveats aside, I think this is a highly exciting area for future work. Okay. So I just wanted to show you one slide, I think it's one slide, that indicates to some extent the importance of these other mutations. So not all the mutations, very few of the mutations that we see in cancer are probably causal. These are slides from people with breast cancer divided by the kind of pattern they have for copy number mutations. So this represents a person who's got a duplication on chromosome 1, a deletion probably on chromosome 11 or 17, I'm not sure, I can't read it here. And these people had simple, small number of changes. These people had wild genomes. And these people had wild genomes, but they were wild in a different way. So these have lots of these events, just lots of them. These have very small finger-like, huge amplifications on particular chromosomes. We call these events firestorms. These patients generally survive, and these patients generally die. So the pattern of mutation is useful for risk assessment in breast cancer. It turns out to be, and this we showed in 2005, it's useful for risk assessment in many types of cancer, including prostate cancer and bladder cancers, among others. So the mutations, even if they're not causal, are useful indicators of what kind of hombre one is dealing with in approaching the patient's cancer. So these are obtainable at very low expense. It is not yet done routinely for everybody with cancer, but it's being done on increasingly common measurement to take in assessment of the cancer's danger level. Another indication that mutations are important came 20 years ago from Bert Vogelstein's study of cancers that have extremely high mutation rates. And these cancers, despite having very high mutation rates, point mutation rates, are generally, they're not benign, but people don't die from them, typically. They're treatable by surgery. One of the theories at the time was that the cancer became loaded with bad protein, and that inhibited its ability to grow robustly. Another theory was that these proteins were immunological targets. The cancer, by making so many bad proteins, was alerting the immune system, and the immune system could fight these cancers down. And in support of that idea is some very exciting recent therapeutic stuff, which shows that that cancers know how to suppress the immune system. And when you can defeat that suppression of the immune system, you can get some miracle cures. Doesn't always happen. It happens. It's dependent on the type of cancer and not in everybody. And these treatments can be also fairly life-threatening. But they are evidence that the immune system does detect that there are abnormalities in the cancer. And these mutations are probably indicating, if we followed these mutations, we would know possibly which proteins are exciting the immune system. So if we go back to this schema, we could ask, given the mutations that are present here, we could ask which of those mutations are being presented on the cell to make them immunogenic targets. So people have not yet, as far as I know, coupled this organoid thing with exposure to the host immune system, but that must be in the works. So there is a mechanism inside of cells whereby, how many of you saw Bananas, Woody Allen's movie Bananas? Okay. Well, remember the dictator asked that you wear your underpants on the outside? Yeah. And, well, that you change your underwear every hour, and for them to be able to check you, you had to wear your underwear on the outside. That was the joke. Anyways, so the cell does that. And the way it does it, it chops up its proteins and wears them on the outside of the cell. That's part of the agreement the cell pays to contribute to the organism. And so the cells get decorated with their proteins, and when the cell decorates itself with a protein that is mutant, there's a good chance that that can be detected as immunological, as immunogenic. And so there's this great opportunity to look at the presentation of those mutated neutral mutations that are nevertheless making these cancer cells vulnerable. If this has the character of stream of consciousness, I'm awfully sorry. That's how I work. Okay. All right. Right. So I'm going to turn to the notion of using these mutations as very sensitive means of detecting the cancer. I'm going to return to this diagram. Cancers kill largely because they disperse in the body. And so I've drawn here, in this hand drawing, various pathways by which cells escaping from the cancer and where they go. So as you probably know, cancer cells sometimes end up in the lymph nodes. It's very important for some cancers that the surgeon takes out the lymph nodes, and the pathologist looks to see, are there cancer cells in the lymph nodes? The cancers also can spread out, and they may be missed by the surgical knife, and the patient can retain at the site cancers that can regrow. This is particularly a problem in prostate cancer. Or they can travel in the same organ to sites that are still so small that the surgeon doesn't recognize that they are there. They can also travel into the blood, and this is something that we didn't know until about 5 years ago with confidence, that cancer cells do appear in the blood, and they can be detected in the blood. And there's a theory which is true at least part of the time, that the cancer cells can come back and home back to the original site. After all, they came from here, and this is a site that they're comfortable living in, so they can go through the capillaries to the bloodstream and then come back at extra, more distant sites within the same tissue. So what I'm going to talk about is being able to detect these things, and you can see that being able to detect these things are going to have some major clinical value. First, I'm going to show you evidence that was derived by Jim Hicks, then and still working as part of our larger group, who showed unequivocally that there are epithelial cells in the blood in people that have prostate cancer, could separate out those epithelial cells, and by doing the single cell profiling that I talked about, could show that some of these cells were cancer cells. This is the profile of, well, this is a bit of a white lie. This is the profile of the cancer, and these are the profiles of individual cells in circulation that could be isolated. Of course, one also isolates normal blood cells with their known genomic markers. So that's evidence that this process happens. Now, clearly, if there is enough signal in the blood, if there are enough cells in the blood or free DNA in the blood, and it's now well established that not only are there cancer cells in the blood, but DNA leaks out so that the blood carries DNA from the cancer. So that there is the possibility, if one had a method of detecting these small events, of knowing, well, has the cancer been removed? Have the cancers in the blood gone to zero? After a period of remission, does one see cancer cells returning to the blood, which would be an indication that there's been a metastasis? One might be able to detect that metastasis before the metastasis is evident radiologically. One might not know where to look. One might be able to then begin treatment on the patient even prior to knowing where the metastasis is. And there's indication, I'm told by a number of oncologists, that if metastases are treated early enough, the patient can survive. So there's this opportunity to detect the cancer if one could detect it sensitively, either the DNA or the cells in blood. And any tool that's capable of doing that would also be able to tell you whether the surgeon left stuff behind, so that the surgeon could go back in and take a larger portion from the tissue if need be. Similarly, I would much rather have a molecular technique telling me if my lymph nodes were positive than, depending on a pathologist, picking out 1 or 2 rare cells in a lymph node. And one can sample the normal tissue distantly from the surgical theater and ask, has there been spread of the cancer in the normal tissue? All of these things and all this information would pertain to prognostication for the patient, the likelihood of their survival, and the degree of heroism that's going to be required in treating the cancer in terms of the strength of the therapeutic regimen that the patient is exposed to. So we've been focusing on this very sensitive detection of the cancer. I showed you that we can do this on single cells already. And I'm going to talk now about, and this is really what my title is named after, this methodology that we're developing. We want to be able to detect these mutations with exquisite sensitivity. What degree of sensitivity? We'd like to be able to detect one molecule from the cancer in, say, 100,000 other molecules of the same type. Now, if you say, well, that's 1 part out of 100,000 for that locus in cells, but that's 1 part in something like 10 to the something like 10 to the 9th DNA fragments total. So when I say exquisitely sensitive, I mean exquisitely sensitive. So let's see what we can detect here. So when you get an inversion or a copy number change, there is, at least theoretically, these bridging fragments that are unique to the cancer. One could look for these in the lymph nodes, in the blood. If it's somebody with bladder cancer or prostate cancer, you could look for them in the urine. Any indication that the patient was in remission or recurring. Detection of these rearranged fragments is actually very robust. It was first done for leukemias. In particular, chronic myelogenous leukemia had a very specific bridging fragment present in 90% of CML. And because it had a very well-defined structure, people used PCR and a variety of other techniques for amplifying. And you cannot make an error doing that. So you can make error-free detection of these with the sensitivity that I described. Doing this for solid cancer is problematical. And the reason is because no two solid cancers are likely to have the same bridging fragment. And finding these in the genome is very expensive. There may only be a half dozen to a dozen of these things in the cancer. And to find them really requires very deep sequencing of the cancer, probably costing in the order of $10,000 to $20,000 per cancer. We wanted to do something that was cheap. The technology would be more difficult than finding bridging fragments, but we would be able to do something that was cheap, affordable, and then run clinical trials to see how useful it is to detect these things. So we want to be able to detect point mutations. As I mentioned, point mutations are hundreds to thousands of times more frequent than these kinds of structural events. Hundreds of thousands of times more frequent. And so you can find them fairly cheaply. So we developed ways so that at the cost of just $1,000 or $2,000, we could find a subset of these. We don't find all of them. We just need to find out, maybe there are on the order of 100,000 in the cancer. We would only need, say, a few dozen. So we don't have to sequence the whole cancer. We sequence the cancer in the normal, and we find these things. Now, the problem with detecting these is that to detect these, you need to do two things. You need to amplify them. That is, you need to grow the DNA in the test tube by this procedure called polymerase chain reaction. So you need to grow them. And then you need to sequence them. And growing them is error-prone. That is, you get more point mutations when you grow them. And when you sequence them, the sequencing has an error rate of 1 in 100, 1 in 300. So your sequence has got errors in it. And that, I think, is what stopped people in the past from pursuing these as targets to trace down the cancer. So I think in close to the last slide, this is a shorter talk than I thought it would take. I'm going to illustrate some of the thinking that went into designing a method to make point mutations robustly detectable, sensitively detectable. And this isn't the exact method that we're going to use, but it illustrates the informatic concept. So you start off with some original set of DNAs in a population, say in blood, and one of them is carrying a cancer mutation that you know because you sequenced the cancer. So you're looking for this. You know to look for this. But there could be 100,000 other molecules that don't have that in the blood. So what we do is we mutate. Before we sequence or amplify, we mutate. We deliberately introduce a known kind of mutation into these template molecules so that each template molecule is distinct. You can tell one from another because a pattern has been imposed on it, a random pattern. Then they're amplified and they're sequenced. And when one sees the G, if it always is occurring in the background of a uniform template mutagenesis, these are, I don't know why these are, because this is too technical. We convert the C's to a U, but the U is interpreted as a T. Don't worry about it. Just think of the U and the T as the same. So we make multiple copies of these, we amplify them, and when all of the copies of the mutant are on the same pattern, then we've outwitted the error proneness of the amplification process and DNA sequencing, because those errors are random. We would see an occasional G maybe in some of these strands, but they would be in the minority of that template form. So that's the method that we are developing. We're expecting to have our first clinical case certainly by the end of the year, in which we're going to explore whether that person's cancer was detectable in her lymph nodes and in her blood by this method. So I think this is my concluding slide. Oops, no, it's not. It's not my concluding slide. I'll just remind you again. So some of the exciting things, I think, in my opinion, are on this slide. One is organoids and the ability of using them to query empirically what the patient will respond to be able to study which subclones respond to which agents and possibly learn what it is that makes subclones sensitive to a particular agent. And to be able to detect very sensitively the spread of cancers within its own tissue and throughout some of the liquid phases of the body, such as the lymph and the blood system, or the urine or sputum. And the impact of this will be to be able to detect that you've in fact operated successfully on the patient, that you have removed the primary and its descendants, or that you have not, and that you still have a job to do. But you can just take that job, you can assume that job at a sooner time before things have gotten further out of control. So in the last slide, I don't know why, oh, that does show. Huh. Okay, so we have had collaborators in mainly the New York area, from Cornell, Weill, Mount Sinai, from Memorial Sloan Kettering, NYU, in Texas, Nick Navin, a graduate student who started the single cell stuff, who went there, and a great collaborator in Sweden, Anders Zetterberg. And these are all great physicians and surgeons, and a very large group of people contributed to the work I talked about. I mentioned Jim Hicks for the single cell in blood, Joan Alexander and Jude Kendall for the work, and Alex Krasnitz for the work in prostate cancer, Hua Wang and Don Levy for developing the methods for sensitive detection, and a variety of people that have helped in a variety of ways, including a graduate student, Robert Abukhalil, who's done some of the phylogenetic work that I told you about. So I have a great group, and we're optimistic, again, about improved treatment, prognosis, and of cancer in the coming years. So that's my talk, a little quick.